Last sessions we talked about cell discovery and cell theory. Okay, we said what is the meaning of a cell. Who can define what is the meaning of a cell? Ahmad Afifi.
A cell is a basic structural and functional unit of all living things.
Okay, Abdullah Ibrahim Yusuf.
السلام عليكم.
Alaikum salam. Abdullah Ibrahim. Yes, what is the meaning of a cell?
Is the basic structural and functional unit of the living things.
Excellent, excellent. Okay, so Ahmad Bajaba. Ahmad. Ahmad Bajaba.
Yes, Mister.
Yes, what's the meaning of a cell?
A cell is the basic structure and functional unit of all living things.
Excellent, yeah, excellent. Okay, Shabbat. So now we talk about the meaning of a cell, and also we talked about the efforts of many scientists when they to discover cell. The first scientist we talked about was Robert Hooke, right? And we said Robert Hooke, what he did, what was the work of Robert Hooke? Sumbul, are you there? You still with me? Go on. Hey. Faris Khalid. Hello. Suhail. Suhail. Everyone, I don't know, you can still hear me or not? Hello? Hello?
We hear you, Mr. We hear you.
I asked, I said names of some persons to talk. Why they are not talking? It means they are not in front of their laptops, right? Okay. Right. So, just to remind you what we talked. We said Robert Hooke, he made a simple microscope and he used the microscope to look at a piece of cork, okay? And he saw small box-shaped structures in the cork. He called them cellular, okay, that we know today as what? Cells, okay? So you have to highlight all of this in your worksheet as I'm highlighting now, okay? I think all of you downloaded it. So the next scientist we said it was Anton van Leeuwenhoek, okay? He used also another microscope. He examined warm water, milk, and other substances. And he was surprised to find he found living organisms in these substances. He found living organisms on these substances. Okay.
Salam.
Salam.
Mr. Page, what?
I'm still revising the page. Watch. You can see the page. I'm sharing the screen. You can see my screen?
Oh, no, I can't.
Everyone, you can see my screen?
We can.
Yes.
And the next scientist we'll talk about, he was Massive sliding. We said he studied what? Alaikum salam. He was Massive sliding. We said he studied plants under microscope and his conclusion was that all plants are composed of cells.
cells.
Okay. The fourth one was Theodore Schwann, and he worked with animals, different kind of animals, and concluded that animal tissues were made up of cells. Okay. The
Okay.
next scientist.
السلام عليكم. How are you, Mr.?
Fine, thanks. You have to listen. The next scientist, okay, was Rudolf Virchow. He proposed that cells divide to form new cells.
Cells.
Okay.
Mister, هل نحفظ should we study dates or no?
No dates, no dates. So Rudolf Virchow proposed that cells divide to form new cells, and we said all of these ideas, scientists agreed to each other to put all of these ideas in a theory. They called it cell theory. We defined it as a fundamental idea of modern biology and includes many principles. Here we have how many principles we have here? We said we have 3 principles. We have 3 principles. Okay.
Sir, we should learn the principle and the explanation, right?
Yes, and you can mention any explanation from your own. Okay.
Can I use my words?
Yes, you can use my word, but not for this one. يعني مثلا the first one, all living organisms are made up of one or more cells. The principle as it is, يعني the principle is يعني it's constant. يعني or if you use a different word but the same meaning, okay. But all living organisms are made up of one or more cells.
So the organisms that are made.
All, he said here, all, don't forget he is talking about all living organisms are made up of one or more cells. Okay?
Sir, can you make a copy of this paper and put it on the
موجود على
website?
الموقع موجود
It is already, Shabab, listen, if anyone asks a question, I'm the only one to answer here. Okay?
Sorry, Mr.
An organism can have one or many cells, and we said plants and animals are an example for organisms with many cells, and we call them unicellular, the animals, or organisms with one cell, multicellular for organisms for many cells. Okay. The second.
Mr. ذا بالphysics.
The second concept, we are now in bio. Don't ask anything about physics now. Okay. The second concept, the cell is the basic unit of organization in living organism. We said, what is the organization inside an organism? We started with what? Who can answer? Basah.
What's the question?
What is the organization? Organization.
It starts from cell and then tissue.
Excellent.
And then yes, thank you.
No, we have 5 levels of organization. We started with cells and tissue. Who can complete?
Cell, tissue, organ.
Organ system.
Organ system, بعدين organization.
No, don't mix between organ system and organism. You said cells, and then a group of cells make what? Tissue. A group of tissues make organ. Here's the word organ system means organ. He's talking about organ. Okay. Then after organ, a group of organs make a system. A group of organs make what? A system. Then many systems form the whole body of the living organism. So in one word, cell, tissue, organ, system, body. Okay? Cell, tissue, organ, system, body. Okay? But the third principle, all cells come from cells. It means cells come from another cells by division. Okay? So cells has an origin from pre-existing cells. The cells, how this happens, we said in heredity, cells pass copies of their genetic material onto their daughter cells. Okay? So everyone, these points that we talked about is clear or are clear for everyone?
Yes, it's clear.
Okay,
and
we'll move. Yes.
The organisms that are made of only one cell are called unicellular.
Unicellular, excellent. And the
Unicellular.
ones that made of many cells, multicellular.
Multicellular. Okay.
Okay. Come to the next topic. We'll talk about microscope. We said scientists use microscope to examine
Yes.
cells. Okay. So we want to know more about microscope. So we have two kinds of microscope. We have the microscope you can find on our lab, which we call light compound microscope. Light compound microscope.
Microscope.
Yes, a compound light microscope. This is the ordinary one we use in our lab. Okay, and the other microscope is the electronic microscope. The electron microscope. Electron microscope. So we have to know some differences between each one of them. One of the main differences, light microscope, from its name. So it uses what? We depend on light. We depend on
Light.
what? On light.
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 But for electron microscope, we depend on what? Electrons. We depend on electrons.
Electrons.
Okay. Right. The second differentiation, what is the magnification? What is the meaning of magnification?
It magnifies the image for
Yes, yes. What is
10,
the
10x, 10
yeah,
times.
10 times.
10 times.
So if you look to the compound light microscope, it is composed of two lenses,
Lenses.
and
So
it
this
Each
has,
lens
which type?
times 10.
and each lens, I'm
Light,
talking
light,
about light here, light microscope,
Sir, which type of
light
microscope this?
Light
microscope,
microscope.
light
Ah, okay.
microscope. Concentrate. I'm not talking about light microscope. We have this is the first lens and this is the second lens.
Lens.
So this lens has a magnification of 10 times, of 10 times. That
Times.
magnifies the object 10 times its original size. Then the
Yes.
second lens magnifies the image from the first lens also 10 times. So it means the object is magnified 10 times 10 equals 100. Equals
Oh,
what?
mister, if there is,
100.
mister, if there is 3 lens, so it's times 1000.
Yes, 10 times 10 times 10. It will be 1000.
3 lens.
But most of compound light microscope uses only 2 lenses because we can have distortion, okay, on the image if we use more than 2
It's enough.
lenses. Yes.
Okay.
But for this magnification, what is the highest magnification we can have from a light microscope? What is the maximum magnification we can have from a light
Times 2,
microscope?
times 20,
Times?
I think. 20
No,
for a lens.
it is 1000. It is 1000.
1000.
The highest, yes, the highest value we can have from a microscope or a compound or light microscope, it is 1000. Here you can see, okay, he said the best compound light microscope only magnify an image about 1000 times, 1000 times. And do you think, who can tell me, this magnification is enough to examine microorganisms?
In the electron microscope, no.
No. Okay. So
But 100,000.
scientists said that we have many organisms we didn't discover that has a smaller size than we see. Okay. So they tried to invent another microscope. They called it electron microscope. Electron microscope. What is the magnification of electron microscope? The magnification of electron microscope can be 500,000 times. 500,000 times. So we use electron microscope to examine virus, to examine bacteria, to see what is inside the cell itself. And by this microscope light compound, you can only see the cell itself, but it is not clear what is inside the cell. But with this microscope, electron microscope, you can see even the small organelles inside the cell.
So what's the largest degree?
We said here electron microscope 500,000 times, but now I think there is a more powerful microscope. Okay, And each country had a different microscope. But this is the most common one you can see now in each country. By this powerful of magnification, you can see it in many countries now, even here in Saudi Arabia. Okay, so if we want to read about light microscope, you see here the modern compound.
Can I read? Can I read?
Okay, read it.
What is a compound light microscope?
Yes.
The modern compound light microscope uses a series of glass lenses to magnify or enlarge an object. When visible light passes through each lens, it magnifies the image of the previous lens. For
Yes.
example, two lenses that each magnify an image 10 times result in a microscope that magnifies the object 100 times, as shown in figure below.
Yes. So now the modern compound light microscope uses what? A series of glass lenses. Okay.
Which is usually two.
Yes. When visible light, so it uses what? Visible light, the ordinary light that we in our room or from the sun. Okay. So each lens magnifies the power of the other lens. Okay. If two lenses, each one by 10, so we multiply the power of the two lenses. Okay. How can we calculate or how can we know the magnification of the compound light microscope? Just to multiply
each lens times
each
10.
lens. No. Yani if I said I have two lenses, the first lens 20 and the second the second one is 30. So 20 times 30 equal 600.
600.
Yes. So each lens could have different power. Each lens could have different power. Okay. What is the maximum magnification power we can have? It's only 1,000. 1,000. Could we have more? Yes, we could have more, but the image or the vision will not be clear. The vision will not be clear. Okay. So everyone, do you have any question about light compound microscope?
Thank you, Mr.
Okay. Now move to the electron microscope. But before we can start electron microscope, I make you show a video about different images that were taken by light microscope and
عندك الدباسة؟
في سوي ميوت.
You can see this video?
Yes, can.
Hello.
You can hear the sound, huh?
Yes.
No.
وين الدباسة؟
ميوت يا
مستر.
عفيفي ميوت.
مستر لو ترفع لنا الفيديو على الموقع واحنا نتفرجه.
Yes, it will be uploaded, but just listen. All of these materials will be uploaded on the website.
This microscope in the school.
يا طيب.
هلا
حبيبي.
تكسين.
عاوز تنقلد يا لما الولد جان لانترو.
يا شباب بالله هدوء، خلينا نركز مع
ايه
اللي بالشاشة.
السلام عليكم.
So these are the parts of the microscope. That eyepiece lens, you use this one to look, okay, and you direct one of these lenses to the specimen or the sample you are examining. You put here on the stage. You put the slide here. Use this clips to fix it on the stage, okay, and use only one of the lenses. So now how many lenses we have? We have this lens and this lens and the eyepiece, okay, and each of these lenses have different power of magnification. Okay, and here's the light come from this one. Okay, this power button, so we can here use a lamp. Here is what a lamp, and direct the light from down up here. It will light up the slide you are examining. Then the light comes from the slide up through this lens and through to the eyepiece lens. Okay, so this is what compound light microscope. Now, if you want to image taken under compound light microscope to see the magnification of light compound microscope. So you can only see the cell, okay? But what is inside, or you cannot see the parts or the fine parts of this organism. You cannot see the fine parts. You can see only the whole cell. See, you can see the cell itself, but what is inside is not clear, okay? What is inside is not clear. So watch it again. So these images were taken by compound light microscope. You can see the cell, but the organelles inside is not clear. Okay, but if we look at different images taken by electron microscope, you can see now the difference. Close your cam.
Yeah.
والله
Stop talking.
مستر وقت الصلاة خلينا بنروح نصلي.
I agree. Mister, I have a question.
Let's wait.
Does plants have tissues and cells?
Okay. Now you saw the difference between the images taken by compound light microscope and electron microscope. You can see a big difference on the clearance of each image. Okay, do you have any question?
مستر ما شاء الله الدرس مرة سهل، فهمنا كل شيء.
So now we can, who has a question?
I agree.
Okay, Shabab. Now we're going to read what is
Mister.
an electron microscope. Rashid, can you read?
Mister, I have a question. May I ask?
Yes, Muhammad, you can ask.
If you ask me what's the difference between two microscope, how can I answer? Yani, what's the good answer?
And try to make comparison. Number one, what kind of lenses they are using?
So I say the light microscope use light lenses.
Uses glass lenses, right?
Yes.
Electron microscope uses what? Electrons, a beam of electron. Now we are going to talk, okay, to read. So you can read.
Yes. The best compound light microscopes only magnify an image about 1000 x or times.
1000 times.
1000 times. Scientists needed more powerful microscopes to learn about cells. The electron microscope was invented in the 1940s. It doesn't use lenses. Instead, the transformation electron microscope uses magnets to aim beam of electron.
So it uses what? A beam of electrons. So electron microscope uses what? A beam of electrons. Okay.
To be magnified. Sometimes can magnify an image 500,000 times. The scanning electron microscope SIM was a further improvement in technology. It produces a three-dimensional image of the cell. One problem with the TIM and SIM is that only non-living cells can be examined. A most recent invention, the scanning tunneling electron microscope (STM) can magnify living cells. Mister, we learned the STM.
Yes. I'm going to tell you now. So what is the main difference between compound light microscope and electron microscope? The light microscope, the magnification. No, just wait, Rashid. What is the magnification of compound light microscope?
He went to Salah, Mister.
Right, Amjad?
1000.
1000, okay. What is the magnification we could have from electron microscope?
500,000.
500,000. And in the video you saw 2 millions. Yeah, an image taken by microscope, it was 2 millions, right? Okay. So what is another difference? A light compound microscope depends on what or uses what?
What? What's the
What?
question?
What is the usage, Brahman Basfail?
Yes.
What type of lens?
It uses what? Beam of electrons or glass of lenses?
What? I can't hear you.
What does it use? What type of lenses?
Compound light microscope. What is the kind? How does it work?
Lenses.
Using a glass of
What type?
lenses. A
Glass,
glass of
yes,
lenses.
Lenses.
But electron microscope using what? A beam of electrons.
A beam of electrons.
This is two main difference between all of them. But here another point is talking about the different kinds of electron microscope. The first one here he said is what? SEM. What is SEM? S for scanning, E for electron microscope. Scanning electron microscope. He said
Sir.
was a further improvement in technology. It produces a three-dimensional image of the cell. So SEM gives us three-dimensional image of the cell, as you have seen some.
3D يعني يا سر.
Yes, 3D. Okay.
Yes,
One
but
problem.
both of them share one thing.
No, they don't
No,
share
both. Yeah, the most common between them that uses a beam of electrons and they can magnify starting from 500,000. But
Mister, they can magnify living and non-living, صح?
just wait, this is another difference between SEM and TM والSTEM. Well, look here. The SEM gives us a 3D, gives us what?
3-dimensional.
3D.
This
توريك
what?
الصورة كاملة.
SEM, yes. But what is one problem with TM and SEM? We have another one, TM and SEM, is that only non-living cells can be examined. Why non-living cells? Why non-living cells? Because the beam of electron will kill any living cells. Will kill any living cells. Okay. So scientists think more deeply to overcome this problem. Okay. They invented STM. They invented what?
STM.
STM. A more recent
STM,
invention.
which is scanning tunneling electron microscope.
Yes, excellent. Scanning tunneling electron microscope. It can magnify living things. Okay, there's a difference between STM and these two. Okay, so we'll stop for this now because I know that you have another section. Inshallah, all of these materials will be uploaded. Okay, including this video. This video is recorded. Okay, so see you inshallah later. See you.
Thank you, Mister. See you later. Bye.
Bye. Don't forget to pray. Don't forget to pray. Yalla go.
يلا سلام.